In one of my first videos, I talked about how cells process information. In this video, we're finally going to start asking how that occurs at a chemical level by looking at proteins that bind to things and then respond in a defined way. We're going to talk over these videos about allostery and cooperativity and things like post-translational modifications. So it's a cool set of videos. I look forward to tackling them. So let's start by thinking about something I briefly mentioned in a previous video, and that is GLP-1, which is a molecule that activates the GLP-1, come on, pen, there we go, that activates the GLP-1 receptor, and then that turns on intracellular things, in this case in beta cells. So a molecule is out here in the bloodstream, binds to a receptor on the cell surface, that information gets transmitted inside, which leads to all of the cool things that, say, GLP-1 activators do. But when you think about it, there's actually something weird or interesting happening here, and that is, if I draw the membrane, which I'm now drawing in high resolution detail, the molecule is binding out here, in this case in purple, and somehow or other indicating do stuff down here. And what I'd like you to think about for a second is how might a protein transmit information from one site out here to a different site, possibly a long way away on the protein. And that's without the molecule actually going inside, just binding outside and transmitting information inside. So think about that for a second. Maybe pause if you want. And what we're going to do is unpack that. So let's quickly talk about the functional cycle that happens in one of these types of receptors. These are a type of receptor known as G protein coupled receptors. And that is universally abbreviated GPCR. So that's what I'm going to do from here on out. We're going to talk about GPCRs in our biology a little bit later. So for the moment, we're just using this as a receptor that does stuff just like many other receptors of different receptor classes. The basic functional cycle of a GPCR is as follows. The protein sits in an inactive conformation. When a hormone or an agonist or something that turns this on binds, it puts it into an active form, shown here in purple. That active GPCR can then bind to a G protein, and that's where the name comes from, this alpha protein, which then triggers a set of events that cause the alpha protein to wander off into the cell, and actually the beta and gamma both. But these proteins wander off and signal, hey, something bound at the receptor. And the part I'm going to focus on for right now is just that inactive to active part, right? That how does information come from up here and go down inside? How does that transition happen from these two different conformations? The overall answer, or the phenomena we're studying here, is allostery, which I'll read the definition. It's the process by which biological macromolecules transmit the effect of binding or change at one site to another. And this site that it transfers to is often distal, meaning distant. And this allows for the regulation of activity. So let's unpack what that means mechanistically in this video. And then in the next video, we're going to look at a bunch of examples to see how it plays out biologically. So how does this work? A good example system on which to introduce this is actually not the GLP-1. We're actually going to talk about the cannabinoid receptor, which is also a GPCR. It's the receptor that is responsible for recognizing THC, so one of the important components inside of marijuana. And so we're going to actually talk about how this receptor works. THC binds to the cannabinoid receptor, and it binds at this top region of the membrane. So this is outside the cell, inside the cell, and it binds up here at the very top. So the THC does not ever enter the cell. Instead, it binds at the top of the receptor. What this does biologically is it activates this sort of neurotransmitter release, and in doing so, it actually overwhelms and desensitizes neurons, which is part of why one feels relaxed when high. It's because basically a bunch of neurons in your cell, in your brain, are kind of in this not quiescent, but sort of desensitized state because of the THC effect. The way that this information is mediated, the way that this actually gets passed on, you see desensitization, is because this receptor has, just like the GLP-1 and just like every other GPCR, an active and an inactive form. And so what I'm actually showing here is a morph between those two structures. So the active and inactive structure, they're both fairly similar to each other. We're not seeing a huge complete rearrangement of this protein, but it is fluctuating between these two states. Now I'm overlaying them on top of each other using the same colors that I showed before, the inactive form in green, the active form in purple, and hopefully what you recognize is it's pretty similar, but that there are changes up here at the top. And there are actually some changes, maybe harder to see at the moment, but down here at the bottom. So these two states differ at both the top and the bottom of the receptor. This G protein I showed schematically, this orange molecule, remember this is what is going to bind to the GPCR and then go off to the rest of the cell and say, hey, something happened at the GPCR, something bound to the receptor. It can bind to the active GPCR. And so this is the structure of a G protein in orange bound to the purple activated cannabinoid receptor. And hopefully what you can see is this helix is kind of sticking up into a pocket that's made here. This pointy diagram was chosen for a reason, right? It's kind of like a pointy top of the G protein, orange, is sticking up into a triangle cleft at the bottom of the GPCR, the receptor. So when this receptor is bound and in the active form, the G protein can interact. But this actually can't happen with the inactive form. So now I'm showing an overlay of the same structure I was just showing in purple and orange, but I've also laid the green structure on top. And hopefully what you can appreciate is even though the green structure is pretty similar to the purple structure, let's actually pause where it's really obvious, right there. Hopefully you can see this helix from the inactive form has folded in, is sort of swooped in relative to what it was doing in the, that's not very clear with the red. It's swooped this direction, closing the pocket and basically taking up the space where the G protein should bind. So basically the inactive form can't bind to the orange G protein. The purple active form can. Okay, so what have we said thus far? We've said that the cannabinoid receptor flips between active and inactive, but that only the active receptor binds to both THC, sorry, my blank disappeared, to THC, remember this is activating with the component THC, and the G protein, that's the orange protein that it sticks to inside the cell. So what we've said thus far, the cannabinoid receptor is flipping back and forth between these two states, and only in the active form can it bind to THC and the G protein. Let's think about this energetically. How can we describe this? Why would adding THC, for example, switch these conformations? So let's start thinking about the initial state of the receptor. If you were building a system here that is going to respond to the addition of a drug, which state would you have favored in the absence of receptor? If you just had these sitting on the surface of the cell, which would you have favored in the absence of THC? So just sitting on the surface of the cell, would you rather favor the green state or the purple state, the inactive state or the active state? So think about that. Which would you prefer? If I were the master of the universe, and this is in fact how things work, this state would be favored. The inactive form would be a favorite form of the receptor. And that makes sense, right? That means that this receptor is going to be off in the absence of the addition of the ligand. If we look at a bunch of molecules in solution, what does this mean? What would you see? Well, it would look something like this. We'd see on the surface of our cell, almost all of our receptors would be in the off state. They'd all be in the green state. Occasionally, one would be in the purple on state. Another one might flicker off, but then they'd flicker on but off, right? So there's a sort of random fluctuation happening. But overall, most of the receptors are in the off form. So what this means is without THC, most, but not all receptors are in the inactive state, right? The receptor is mostly green in the things that I've shown thus far. Cool. Now imagine that we add THC. THC is going to like to bind to that version of the receptor. And what we're actually going to do is stabilize this. It's going to become more and more common. It's going to become the common state of the receptor, which means if we looked at the entire population, if we started adding THC, it's mostly green with an occasional purple. Now the receptors are turning purple that have bound to THC. we add more THC, we add more THC, and overall the entire system switches from being in the green inactive form to being in the purple active form. And this is an example of Le Chatelier's principle, which is where basically you change the concentration, usually in biochemistry, of a system, and then shift the equilibrium position of the system to counteract that change. So you basically, if you add a bunch of reactants to a reaction, it's going to shift the equilibrium to favor products. If you add a bunch of products, it's going to shift the equilibrium to favor the reactants. And in fact, if you, this is one of those verbal question things, if somebody asks you a verbal question on a biochemistry exam, the Chatelier's principle is 90% of the time going to be part of the answer, right? So I actually tell students when they're taking a class of mine, and if you're taking a class of mine watching this video, you'll hear this right now. If you don't know the answer to an exam question, just write down Le Chatelier's principle. It's a good first guess. And so let's see how this plays out in this system. So without THC, remember, most but not all the receptors are in the inactive form. So the receptors are all green. But with THC, most but not all receptors are in the active state. So we're somehow able to drive this transition. So how did this happen and how can we describe this? Well, we're going to describe this using the idea of linked equilibria. So we've already seen lots of equilibria or individual equilibrium in this class where, say, we've got something, we've talked about a molecule that's bound to something, that thing falls off. So you've got sort of a dissociation reaction. That's what this would be here. And then we've got another equilibrium here where purple and green are in equilibrium with each other. But with linked equilibria, what we're doing is we're linking these together. So basically the products of one reaction are the reactants of another. And I'm going to draw this schematically this way or with sort of math symbols. So we're going to talk about I, which is our inactive form. We're going to talk about A, which is our active form. And then we're going to talk about X, which is for this particular receptor is THC, but it's whatever small molecule binds and activates this receptor. All right, so now let's walk through what happens with these linked equilibria. Oh, here I have the definition here. It's two or more reactions with the products of one reaction are reactants for the other. So that's the definition. Okay, so let's think about what's happening in the system. So in the absence of THC, we said that this receptor is going to favor the inactive form. So it's just going to be sitting there, and most of the time, most of the molecules are in this form. Occasionally, we're over here. Now, we add THC. What is it going to do to this equilibrium right here? Just if this was all that happened, the answer is it wouldn't do anything at all, right? Because neither of them interact with THC. So when I add THC, I'm basically adding it to both sides of the reaction, right? I'm adding, because it's not interacting, there's no difference. So I can add more and more and more THC and nothing happens. But this system over here, remember the active receptor or the active form can bind to THC, which means that when we add yellow here, we're actually adding products to this dissociation reaction. So we're adding excess products, adding lots of products. And Le Chatelier's principle says if we start adding to one side of the reaction has to shift this way to accommodate that, right? So we've added more and more and more reactants, which means we're going to shift, sorry, I said reactants, these are products, more and more and more products to this reaction, and that's going to shift the equilibrium to favor the reactants. So if we continue to add more and more THC, what we'll actually see is the entire system drift. Oh, wait, there's one other part. So, and as the products of this reaction start turning into the reactants, we're now depleting the reactants for this reaction, right? And so we're going to start seeing green come into purple to replenish the purple that was lost. And if we run that long enough, we're going to end up all the way over here, where basically the entire system is in this bound form. Let me restate that because it's a little bit complicated sounding. There's 4 steps we have to worry about in an equilibrium like this. First, we're adding excess X, and what that's going to do, and that's going to add excess products here, right? So we're going to be, this is 1, we're adding excess products. This is going to shift the equilibrium, and so we're going to drive this equilibrium in this direction too. And that's because we're adding products, so we need to sort of shift to favor reactants to balance out the system. But as we do this, we're depleting the reactants of this reaction, right? Of A going to I. That's being depleted. Le Chatelier's principle says as we deplete the reactants to a reaction, we have to change the products. And so we're going to see this shift in this direction. We're basically going to see more I being formed. So what adding X is doing as it binds to A, it depletes A, so I has to flow into A. And if you do that long enough, you end up with all A. And what we're seeing then is that Le Chatelier's principle is driving the equilibrium from being favoring the, wow, I drew that totally backwards. Inactive to active. Glad I looked at that as I was talking, or I would have just had a very confusing video. So the Chatelier's principle drives the equilibrium from inactive into its active form. All right, so let's talk about the key ideas here, and then in the next video, we'll unpack some specific examples of how this plays out. So allostery is when the change at one site alters the activity and binding at another site. And this allows a protein to respond to a signal. In this case, the GPCR is signaling, hey, I bound something to the rest of the cell. The ingredients of allostery are that the receptor or the protein populates more than one state, in this case, inactive and active. Those conformations have to differ in their activity and the affinity for the signaling molecules, right? If they bind to the signaling molecules the same, you won't see an effect. If they have the same activity downstream, you won't see an effect. And I think I'm going to have to make myself disappear. Last one. We can describe allostery using a linked set of equilibria. Okay, so that's the basic idea of allostery and linked equilibria. In the next video, we're going to unpack some set of examples. So what are the sort of stereotypical features of this, and then how does this play out for a few different real biological proteins. So looking forward to doing that with you. See you later.